So far this semester, we've talked a lot about DNA and how DNA is the instructions for life. And so in this chapter, we're actually going to get into how exactly DNA gives us instructions for life. In other words, how do these A's, T's, G's, and C's determine our traits, like the color of a flower, or the color of our eyes, or how tall we are, or how good at sports we are. And so this process, the connection between genetic material and our traits, was first figured out by scientists that realized that DNA somehow connected to enzymes. And they were looking at basic biochemical pathways, kind of like what we saw in cellular respiration, and noticed that you can inherit, so notice inherit is the key word, problems with enzymes. And so that led them to realize that there was a distinct relationship between genes and enzymes. And so this was called the one gene, one enzyme hypothesis, a direct relationship. If you inherited a problem in one gene, then you would have that same problem in a corresponding enzyme. And so they realized that the DNA leads to specific traits by controlling the production or synthesis of enzymes and more generally proteins. And this process is called gene expression. So this is the expressing of genes in the DNA. So you have your instructions. When you read those instructions, the readout of that is the protein, and the protein is what leads to traits. So as I mentioned, this was originally called the one gene, one enzyme hypothesis. Today it's referred to as the one gene, one polypeptide theory. And the reason why it was modified is because enzymes are just one type of protein. So remember, protein is a very broad category. It includes things like enzymes, but it also includes things like structural proteins. So for example, collagen, as well as a whole other subset of proteins, which hopefully you remember from that table back in chapter 3. But even, so one gene leads to one protein, but even more specifically, one gene leads to one polypeptide. So for example, the protein hemoglobin, so hemoglobin is a protein that's important in your red blood cells, but it's a protein that's actually made out of 4 different polypeptides. So this is quaternary structure, again, sort of referring back to chapter 3. Their multiple polypeptides will form a functional protein. And there are multiple genes that are used to make hemoglobin. So basically, hemoglobin actually has more than one gene that is read in order to make functional hemoglobin. So that's why we now refer to it as the one gene, one polypeptide theory. So the idea of gene expression, so again, still referring here broadly to gene expression, is really about the flow of information. So a gene is expressed when the information in the gene is read, and it's going to be read in a very specific way. And this is called the central dogma of biology. That's how important this concept is in modern biology. It tells us that information is stored in the DNA. That's the base of all information in the cell. It's stored in the DNA. Then that information will be converted into RNA. So the flow of information is from the storage unit, the DNA, to the RNA, which is a working unit. And finally, that RNA will be read to make protein. So this is the store of information. This is the working copy. And the protein is the final product, or the product. So why is the cell going to have this sequence? Why doesn't it just go straight from DNA to protein? Well, let me give you an analogy. Think about when a building is being built. The architect designs the building on his original blueprints that he makes, right? And then those blueprints are going to be used in order to make the building. But think about it. Is the architect going to give his only copy of that blueprint, that store of information, to the workers, to the builders? Is he going to send that out into the rain and into the snow, out on the work site? No, because he wants to keep his stored copy of information in pristine condition. He wants to keep that perfect. So we have our blueprint that's the store of the information, and then we make copies of that information, and we give the copies to the workers out in the work site, and they use the copies to make the building. But as long as, if something bad happens to the copy, if the copy gets damaged or messed up or something like that, I still have my perfect copy back in the architect's office. And so again, that's what's happening with DNA to RNA to protein. I have my perfect thing, my store of information, my blueprint that I want to keep perfect in the nucleus. I'm going to make working copies of that information, which is the RNA. The RNA is what goes out, and the builders read the RNA to make the product or the protein. These processes, so this step here and this step here, are two different distinct processes we're going to be talking about in this chapter: transcription and translation. So this first one here, DNA to RNA, is transcription. The second part is translation, which is RNA to protein. Notice that these names have to do with language terms. So transcription is, say, when like a court transcriptionist is listening to what is being said in the court in English, and they type that into the written form of English. So transcribing is really going from one form of the language to another form of the language, so from spoken English to written English. But translation is when you change languages. So going from English to Spanish is a translating between languages. The same thing is true with DNA, RNA, and RNA to protein. When I go from DNA to RNA, I'm in the same language, just a different form. Transcription stays in the language of nucleic acids. Translation is translating from the language of nucleic acid to protein. This is a totally different language because remember, nucleic acids are made out of nucleotides. So I'm taking one nucleotide language and transcribing it to a second nucleotide language. But when I go from nucleic acid to protein, I'm going from a nucleotide language to an amino acid language. And so this is going to be a very different process. In prokaryotes, transcription and translation will both happen in the cytosol. Why? Well, remember, prokaryotes don't have a nucleus, so everything will happen in the cytosol. In eukaryotes, remember, we have the nucleus, so transcription will happen in the nucleus, and then the RNA will go out of the nucleus so that translation can happen out in the cytosol in the ribosomes. So in eukaryotes, transcription is in the nucleus, and translation is in the cytosol where the ribosomes are. Eukaryotes also have an extra step. So eukaryotes will go from DNA to something called a pre-mRNA. Then they'll make a final mRNA or a mature mRNA, and that will be made into a protein. So notice not just the transcription and translation, so that's here and the last step there, but they have this extra step here called RNA processing. So RNA processing is an extra step in eukaryotes in between transcription and translation. And we'll talk more about all of these coming up in later videos. So I just want to end here by connecting back to this idea of traits. So this is an example of how the DNA, so remember the gene is within the DNA, is copied into an RNA, and that working copy of RNA is read to make a protein, and then that protein will lead to traits. So this is just an example in the fruit fly, which is a common scientific model. And fruit flies can actually have red or white eyes. And this is a genetically controlled trait. So if you have one gene, then that gene will make the protein that allows the eye to have red eye pigment. So the protein is important in making red pigment. In the wide-eyed fly, they have a mistake in their gene. So it's the same gene, but with a mistake. Because the gene has a mistake in it, I don't get the RNA. If I don't get the RNA, I can't get the protein. If I don't have the protein, then I can't make the red pigment. So I don't have red. So their eyes appear white because they can't make red pigment. And again, the reason why they can't make red pigment is because there's a difference in the gene, so that they can't make the RNA, can't make the protein, and the protein's needed to make eye pigment. And that's actually how a lot of the traits work, even in our own traits. For example, skin color. We have differences in the genes that cause the different tones of skin color. So if I have less of those genes, then I'll have less skin color. If I have more of those genes, I'll have a darker skin color. So same idea across all organisms. And that's the central dogma of biology. It's so important to modern biology.